Wallwork or Warwick?
Wallwork.
Wallwork, who's visiting Iannetti's lab at UCL. She's based, you're in your second year of your PhD?
Third year now.
Third year of PhD. Is a guy called Lorimer Moseley. If anyone's come across Lorimer's publications in pain and cognition there, they're always quite different, aren't they, in terms of Lorimer's view of pain, basically. So Sarah's spending 6 months in Iannetti's lab, working on an observation of a blink reflex and peripersonal space, which is an indication of, well, you're going to tell us what the indication was. So, Sarah, thank you.
Cool. So I guess I'll probably spend most of my talk about this defensive peripersonal space, but I also wanted to give you a bit more of my background, just so you know kind of the way that I sort of approach this sort of thing, because it's a little bit, as I said before, it's a little bit different to how Giandomenico has approached it. But yes, my main supervisors are Lorimer Moseley and Giandomenico Iannetti. So first of all, probably tell from my accent, Australia. This is where Adelaide is. If you don't know, we always say down the bottom in the middle. We've got lots of wine, great beaches. I can sell it. So I'm a part of the Body and Mind Research Group, headed by Professor Lorimer Moseley, and we're at the University of South Australia. So like Lorimer, my background is that I'm a physiotherapist. And I guess partway through my undergraduate, I was exposed to a bit of research, particularly with Lorimer, but also with another guy called David Butler. And he's also a physiotherapist, and he's sort of got a business that sort of bridges the gap between research and the clinician, so trying to take it out to the patients a little bit more. So my previous research had been in left-right judgments or graded motor imagery. I don't know if any of you are aware of or heard of this. A little bit with illusions as well, such as the rubber hand illusion and complex regional pain syndrome. So now in my PhD, it's more of a neurophysiology, which is obviously quite different to what I was looking at before. So it's quite new for me. So I'll start with a little bit of my previous research. So when I talk about left-right judgments, we're talking about looking at a body part and trying to say, is it belong to the left or the right side of the body? So if I present to you a couple of images, I want you to, you can say it out loud, but say if you think it's a right or a left limb.
Right.
Cool, so it can be actually quite tricky. And the process of trying to determine whether it's a left or a right limb basically requires on what we call like intact body maps of that limb. So when you were looking at those images, what you do in your head, you may or may not realise it, but you mentally rotate your own, say, hand from the current position that it's in to match that of the image that you've just seen, and then you either say, yes, that's correct, or no, that's not correct, and then you rerun that process again, but for the other limb. So yeah, as I said, it requires an intact body schema, and there's quite an accumulating amount of evidence now for a relationship between some complex pain disorders and distortions of these body representations. And hence we've seen that the response time and the accuracy to determining a left or right limb can be found in people with pain. So this has then led to a treatment called graded motor imagery that Lorimer's had a lot of work with, but also David Butler. So basically, this form of treatment is more sort of addressing the neural maps in the brain for these body parts. So the first task that this requires is an implicit task of the left-right judgments. So they continually go through, is this a left or a right? If you change the orientation of the hands to be in more complex postures, they find it a lot more difficult. There are some people, particularly with like CRPS, who even find this task challenging or even pain-provoking. The next component is what we call imagined movements. So that's an explicit task. So you mentally think about where your current hand is, you look at an image, and then you mentally rotate your own limb to match that. And then the final one is mirror therapy, which is quite well known for things like stroke as well. So there's a bit of accumulating evidence now for graded motor imagery in some complex pain states, particularly CRPS is where the literature is. So research has then gone from this disrupted body schema of that limb to a disrupted space around that limb. So one part of evidence is this experiment that Alberto Galacci did, where they found that by crossing the arms over the body midline, they found that there was a reduced perceived intensity of both noxious and non-noxious stimuli. And they thought that that was based on the conflict of somatotopic and space-based frames of reference. So normally your right hand resides in the right side of space, normally your left in the left side. As soon as you cross over that midline, there's a conflicting thing up there. So there's a very new study that hopefully will be coming out soon that's looking at the disrupted spatial processing in pathological pain, and it's biased towards the healthy side of space and is confined to body relevant stimuli. So basically what we've been looking at is that in some patients, particularly with CRPS, is what this study was on, they show some signs of neglect, but not the classical neglect that you often see in stroke patients. So often in the stroke patients, if you ask them to draw a house, they might only draw half a house. And if you ask them, what's wrong here, they can't pick up on this because they neglect the whole right side. So we were wondering whether, this neglect was perhaps still evident in people with CRPS, but perhaps is it body specific? So this, there were 3 experiments. The first one was a left-right judgment task, which was the body relevant, and a letter orientation task, which was body irrelevant. There were 2 temporal order judgment tasks, 1 tactile, 1 auditory, and also 2 line bisection tasks. One was done on the arm, and one was just the classical one done on paper. So in this first task, we asked them to do left-right judgments of limbs. So first of all, there was an affected limb and an unaffected limb. And then the image was shown in the affected side of space, on a monitor in the affected side of space, and unaffected side of space. And I'm sorry if this gets confusing, because I find it confusing as well, just because you've got healthy space and healthy limbs. But basically, what we found was that there was a bias for left-right judgments for the body relevant. So when the image, there's an effect of limb where people were less accurate at their it took longer to identify their affected limb than their unaffected limb. And if it was taken into their affected side of space, even if it was their healthy limb, they also took a lot longer to recognise it. So basically there was an interaction here. So you can hopefully see on the far ones, over here. So there was an interaction. So this is the unhealthy or the affected limb, and this is the unaffected limb. So here this is the healthy limb in the affected side of space and the healthy limb in the unaffected side of space. Is that clear? So it was dependent on where it was. So that was then contrasted to a letter orientation task, which was also presented in the healthy and the affected side of space, and there was no difference. So the second task was tactile temporal order judgments. So we had obviously on the affected and unaffected limbs, and so we found that stimuli had to be presented roughly 25 milliseconds earlier to the affected hand for it to be considered equal to the unaffected hand. When we did auditory temporal order judgments, which were done right next to the affected and unaffected hands, there was no spatially defined bias. Then in the third task, we did the line bisection. And basically what we found was that on the classical paper line bisection task, so when you have to cut it in half, there was no difference. Whereas if you drew the line on the forearm and then had to do the line bisection task, there was a bias towards the healthy side of space. So basically you can see, hopefully from this, that these changes of a disrupted body representation also start to go in towards the space around the unhealthy or the affected limb. So this got me quite interested in the idea of, how do we perceive the space around us, and how does that relate to, I guess, our body maps as well. So I started looking at this idea of a defensive personal space, which Giandomenico Iannetti has been doing some research in. I will quickly go through what it's basically, how we measure it, what it's about. So it's this idea of a protective boundary around us from the world, and we have been measuring that using reflexes. So the hand blink reflex is an electrical stimulus, non-painful stimulus to the median nerve at the wrist, and we record an eye blink response, which is about 45 milliseconds after the stimulus is delivered, on the orbicularis muscle.
You get a pupil response, by the way, as well, that's quick enough to measure.
A pupil response?
The pupil constricting or dilating. We look at that.
I haven't looked at that, I have to say. Possibly. I'm not sure if that would be...
Well, the eye blinks would be the enemy of measuring any sort of pupil dilation.
Well, they're very rapid, these eye things, aren't they? And obviously the pupil.
Yeah.
Yeah, and I'm not sure if a pupil would be a
arousal,
defensive type. Yeah, around, yeah,
quite
maybe.
quick.
Yeah.
I don't know, it might be worth looking at.
Yeah. So with this reflex paradigm, what Giando has found earlier on is that when the stimulated hand is within the peripersonal space, you have a much larger blink or much larger defensive reaction compared to when the hand is further away. So just as an idea, this is what our data looks like. So you can see the blink starts at roughly 45 milliseconds after the artifact, and you can see that it's quite clearly a lot bigger when the hand is close to the face. So I've been doing, I guess, some fundamental experiments just to try and understand a little bit more about this paradigm, seeing we don't know a lot about it. So the experiment that I presented in Buenos Aires at IASP was this experiment here. So we were asking how quickly can this top-down modulation of this reflex occur. In previous experiments, what they've done is they've had the hand in the peripersonal space for 30 seconds before they receive the stimulus. So they ask them to go into the near position, they get the stimulus, then they ask them to go into the far position, wait 30 seconds, then they get the stimulus. But we wanted to know whether, if you have your hand there for, say, 2 seconds, is that enough time for you to be able to realise that the hand is there and still have this upregulated response? So the way that we did this is we still had a constant 30 seconds between each stimulus, but we just asked them to move their hand at an appropriate time so that their hand was either there for 2 seconds, 5, 10, or 30 seconds before they received that stimulus. So we were predicting that there would be no difference across the board. We expected that at 2 seconds they would still be able to get this upregulated blink. And this is what we found. This is quite confusing, so I'll break it down, but as a very broad overview, the blue and the red at the top there clearly seem to be the largest responses. So the solid colours are all when the hand is near, and the dotted responses at the bottom is when the hand is far away. So I'll break it down. So hopefully you can see that, but the top one here is a main effective hand position. So regardless of how long the hand had been there for, it was near and far. So the blue is when the hand was near, the red is far. So you can see that there's quite a clear difference in averages that between those two conditions. Sorry, this is for 17 subjects as well. We also found that there was a main effect of time. So I'll explain what the graphs underneath are. So you've got the blink at the top. This strip here is the F value. So basically the analysis that we've done is a point-by-point analysis. So what it does is it measures or compares, so you can do ANOVAs or t-tests, and it measures at each time point. So under the big solid colour rectangle over the top is where, on our point-by-point analysis, there is an effect. So outside of that, there seems to be no difference. And this is the corresponding F value for that significant period. So we also generally have a consecutivity threshold. So that, I guess, rectangle has to be at least 10 milliseconds long to account for the multiple comparisons. So you can see in the top one, there's a clear effect there. In the bottom one here, however, where we've seen an effect of time, even though you can see there are lots of little rectangles there that cover quite a lot of the waveform, none of them actually met the 10 millisecond consecutivity threshold. So according to that, there's no difference in time. However, when you do an area under the curve analysis, it finds that there is an effect of time, and it's between 2 seconds and 10 seconds. So what we found is that when the hand was there for 2 seconds, there was a greater response than when the hand was there for 10 seconds, which wasn't necessarily what we were expecting, but I think that it could be perhaps an element of surprise that while it's only been there for 2 seconds, you aren't really quite sure about it, what's happening, whether you're going to get the stimulus straight away. Whereas after 10 seconds, you've sort of been holding your hand there for a little while and, you know.
Did you try this in a block design and the event-related design? So have you tried it with maybe just a design where they make a movement and they get the stimulus, they make a movement, they get the stimulus, so it's not the same, because that would then get rid of this element
of surprise. Yeah, no, I haven't.
And the other thing is, where are your participants looking? Are they fixating on something?
Fixated on the person in front. Yeah. So yeah, so that's what we found. So based on those results, we then ran another experiment to see, can this modulation happen immediately? So as the hand is moving, and does it even possibly have the ability to predict a potential threat? So the way that we did this one was we had the arm was constantly moving. Sorry, I have to say we had 3 different conditions. One where the hand was not moving, just like our previous experiment, and then we had one where people were moving their hand at a constant rate to a metronome, and we stimulated the wrist in both the near and far conditions while the hand was either moving towards the face or moving away from the face. So we expected that, yes, they'd still be able to get this upregulation straight away, and if they are able to predict a threat, that in the moving towards the face, it would be upregulated again, just because having that threat moving towards you can be a little bit startling sometimes. So again, this is a little bit confusing. I'll break it down again, but this is what we found. So the solid colours again are near, and the far colours are far away. So if I break it down first to the effect of movement, so here we've got in the green, we've got no movement at all, so stationary hand, and then the blue we've got the hand is moving towards the face, and the red we've got the hand is moving away from the face. And I think we didn't find an effective movement at all across the board. You can see that there's a few random parts where there is a difference, but overall there was no effect. But what I think is really interesting is the shape of those waveforms, because it's quite obvious to me that the green one has the tail comes down a lot quicker, but we didn't find an effect. So, interesting observation. Then when we compared the towards and away, we found something quite interesting. So again, you've got the solid colours are near and the dotted ones are away. And it looks like you can't see the far towards the face condition, but it is there, but it's just a little bit higher. So I'll break this down again. So in the top one here, we've got movement towards, solid is near, dotted is far, and there is no difference. And you can see there's a couple little points at the end, but really there's nothing there. They're the same. However, in the moving away, there is a difference, and it's quite a substantial difference there as well. So what we think this suggests is that, yes, it can be upregulated quickly, and that it possibly even has the ability to predict the threat coming towards the face. Because in that, when it was far away but moving towards the face, it was upregulated as if it was already next to the face. So we found that quite interesting. So I guess in terms of mechanisms, there is a hypothesis, which is yet to be proven, is that there's a tonic top-down facilitation from the cortex of the interneurons in the medulla. They think that this facilitation is from frontoparietal areas representing the peripersonal space. But this is a hypothesis that remains to be tested. So I guess other projects that I'm working or finishing on at the moment is I've been doing a systematic review and meta-analysis on the effect of clinical pain and defensive reflexes. So this is the idea that, so we've got 3 different reflexes that we've identified as being defensive. One is the startle, one is the blink, and the other one is the nociceptive flexion reflex, or the withdrawal reflex. And we just wanted to see, I guess, test the hypothesis that people in pain have a heightened defensive reaction, perhaps like a protective reaction for a vulnerable body part. The other project that I'm working on is using virtual reality. So it's still using this near-far paradigm with the reflex, but what we've been doing is we've been getting them into a virtual world. So they wear the Oculus headset and immerse them into this new world where they get a new arm, and basically we manipulate what they see so that their proprioceptive feedback is different to their visual feedback, and seeing how that changes, I guess, the blink component. And then a more sort of, I guess, neurophysiology study that I'm doing as well is looking at the recovery cycle. So looking at the trigeminal blink reflex, which is where we stimulate on the forehead, compared to the median nerve stimulation, just to see whether they use or utilize similar pathways. So by looking at their recovery cycle, helps us to understand a bit more about that. And that's me.
Any questions for Sarah?
Was it always in the line in the eyesight? So they could always see the hand?
In the experiments that I did, yes.
Okay.
Although Giando has done experiments in the past with the eyes closed,
Yeah.
and there was still the upregulated blink.
Yeah.
Although when they did another study where they had their eyes open but had a board,
Yeah.
occluding the hand,
Yeah.
they found that whether the hand was far away or near, there was no difference. So there was no upregulation once they had that board, or perhaps a protective barrier
Yes.
between the hand and the face.
So it seems like it's almost like a threat response that's being modulated by the visual feedback.
Yeah. Which is sort of why I was interested in doing that virtual reality study, because there was, I think Giando had one thought that by having this headset, that'll act as a defensive barrier and that you won't even get the upregulation. But I found that there was still an upregulation, but it's a somewhat, I haven't finished analysing this yet, but they still seem to have that.
You still find it, then that's pushing more for that visual side.
Yeah, but I think so it obviously needs some kind of visual, well, it relies on some visual if it's there, basically. Yeah.
This whole area of proprioception, though, is fascinating in terms of how fluid it is. And you've shown that before, it's been shown in the rubber arm illusion, isn't it? It's not, it used to be seen as a purely visual illusion. You have to see the arm. But of course you don't. You get the proprioceptive drift, which is irrespective of what you see. So, I mean, pain populations are always fascinating to work on, in some sense, experimental models. But what the hell is peripersonal space? We asked you this before, but what is it? Is it some aura?
Well, yeah.
I know Riki did a lot of work, didn't he, with rakes and recording from visual fields, that the rake extended the...
Yeah. So there's been a bit of research in like tool use, and the idea that using a tool extends this peripersonal space. But I think the literature is a bit more conflicting on that now, because I think, whether it, some people say it extends it, some people say that it's still around a hand, but then the tip of it, or whatever it is that you're manipulating with that object, is then included in that.
There's something I came across with monkey recording, which I think might be quite pertinent here. They're recording from some somatosensory areas in the monkey, or some area of representation of somatic. If that monkey's hand is just lying there doing nothing, nothing's going on. The second it's engaged in action, the whole bloody thing lights up. So that's fascinating. Because the hand's doing nothing, I think even touching it there was no response. But as soon as that arm was engaged in a voluntary, meaningful action, all this architecture, neuronal architecture, became online. So there's something in that, in terms of this space being only there when there's...
But I mean that could also be, well, I was going to say attention, but I guess there's no response to tactile and touching, maybe perhaps not so much. But yeah, interesting.
Do people who are blind have this kind of peripersonal space that causes a reflex?
Yeah, interesting that you asked that actually, because I didn't include that on here, but that was another one of the studies that I've been working on. But unfortunately, the population of blind people in Adelaide is quite small. So I collected 8 people's data of people who were totally blind to see if they had this reflex, or how many people had this reflex and did they still have this modulation. And out of 8 people, I got 1 that had this reflex, so that's a lot less than normal. And there's 1 person that I found that did have the reflex. I didn't see any modulation. But whether you would see it in 1 person, usually yes, you probably would, because it's quite a large effect. But I don't really know, to be sure. But I think it's interesting though, because I spoke to quite a few of them about, this idea of protection, and a lot of them almost seem to have a heightened protection because they were
blind, really.
Yeah, well, they were quite worried about branches hitting them in the face. They'd often wear glasses because when it was windy, they would often get dust blowing into their eyes. So they seem to, but whether it's shown in this paradigm, I don't know.
It's kind of a learned thing as well, isn't it?
Sorry.
So I was just going to ask whether you were all blind participants, have they all been blind from birth, but some of them required sort of become blind later?
They were all. I couldn't find anyone who was.
You want a congenitally blind population?
Yeah, the closest that I found was someone who was blind at 2, and she was actually the person that had the reflex.
Critical period involved there. David?
I've got a number of questions. One which was actually just a follow-up, which I thought that I'm wondering if you attached something, another sensory cue while you were doing it to the blind population, whether or not you might get that. So if you attached something that was making a slight noise, or something you would give some other cue as to the location, whether or not you might get to peripersonal space within a blind population, because they'd be more familiar with sensing things using those other senses.
You mean like an auditory sound?
Yeah, not auditory sensation, so something which gives a clue that something's getting closer. Whether or not you might get that, or even if you'd need to have them move the limit, if you had a sound that was getting either louder or quieter, or sound as though it was coming closer or further away, whether or not you might get a similar effect even with a non-moving limb.
Yeah, that would be interesting. I think the problem in it is how you measure that, or maybe if you have like constant noise as your hand comes back and forth, and then you give them the stimulus.
Yeah.
Still
Yeah,
something
maybe.
like startle response you'd expect it to make possibly.
Yeah.
If someone's startled by a sound, they'd still have that.
You've got this Ethan's coffee thing as well. If you passively
Yes.
move that person's arm without them doing it, that will then.
I'm not sure, we haven't tried that, but I think that's a good point as well. I think that
To try that with your movement as well, see if there's any sort of representation or momentum effect. So if you have someone's arm manipulated automatically, if you actually only move it here but you move it faster, do you start to get the interesting effects you were getting when you were moving it towards the face without actually even needing to get it further towards the face? It's just literally the brain.
I tell you what to do. Once you've got the rubber hand in body ownership, move that.
Well, yeah, that's what we tried doing with, in a way, I guess, with our virtual reality. But
Yeah, of course. Yeah.
yeah, I think with virtual reality, though, I think because you get a potentiometer at the elbow, and that's how we measure where their hand is in relation to their face. But unfortunately, with those sorts of things, when you're always going to get tactile feedback unless you use like lasers or something else. So I think there's always that component to it as well.
I was also just wondering about whether or not you could look at an attentional manipulation or something like this, so having them perform this task while performing some kind of auditory continuous performance task, whether or not if they're distracted from the motion that their hands make, whether or not you might attenuate so how much of it is going to be something physiological, how much of it will be intentional mechanism.
Yeah. I mean, as I said, like these are all fundamental experiments, and there's so many ideas of ways that you could manipulate it to try and understand it a bit more.
Do you think it's the point of a bigger or a smaller blink reflex? Because normally blinking, the role of the blink isn't going to change, it doesn't matter how big you blink. I mean, have people looked at what the maximal possible blink reflex is and why one would happen?
Well, one thing I know is that it's quite variable between people. So someone who's got a big blink here compared to another person, it can be totally different. And I think that there is a ceiling effect as well. I think that once it gets to a certain height, it seems to be, from what we've found, that you can't really get it any higher. So
Because adaptively, there's a point where that kind of startle reflex stops being adaptive, because although it may be in some way adaptive when something aversive happens to protect your eyes, for example, by blinking, it gets to the point where you don't want to come close to it and longer for something aversive which potentially happening. I'm just really interested in whether people really understand. Obviously, you've got a modulation of a reflex, but really what that reflex is doing.
yeah. Well, I guess we probably don't know that much about that necessarily, but we find it's defensive. We predict that it's to protect the eyes.
Yeah.
But it's just like, so in my review I've looked at some other reflexes. So one has been the withdrawal reflex, which is the classic idea of touch a hot plate and you withdraw.
Yeah.
But the one that I guess they use more scientifically is they stimulate the sural nerve at the ankle, nociceptive stimulus, and you get a flexion of the knee, withdrawal type response, and that's been interesting because that's, I guess, also a defensive type reaction as well.
And you can measure a sort of amplitude of that response.
Yeah, so you can measure it with EMG, but classically with that reflex, people seem to look more at the threshold. So at what intensity do you need to stimulate it
in order
to be able to elicit that response?
Yeah. That's really interesting.
Right. Onward. Thank you, Sarah.
Thank you.
Rating.